All right, so my slides went away for some reason. They were here. There they are. So I'm going to take the baton from Abul, who told you how T cells get activated, and think back to the lymph node that he described and all those events that involved co-stimulation and antigen recognition and the generation of a clonally expanded population of cells. And I'm going to focus on CD4 T cells after they're differentiated and become effector cells that can actually do something useful, but also may be able to do things dangerously, like causing autoimmune disease. Shiv, this afternoon, is going to talk about CD8 positive T cells, CTLs. So some of the questions you had about CTLs killing tumors, the mechanisms by which CTLs work, you'll hear this afternoon. Okay, so again, a large topic, and the way we're going to focus and narrow down here is focus on CD4 T cells that come into different flavors, different subsets, based largely on the types of cytokines they produce. I'm going to give you an overview of how you get these T cells out of the lymph nodes into the sites where there is infection, and then describe that not all CD4 helper T cells are the same, and we'll describe these subsets and their relevance to defense and infection. And again, try to intervene in various places in the lecture about how this knowledge helps in designing therapeutic strategies, and some that are already in place. Okay, so this is the life history that Abul talked about CD4, CD8 positive T cells from the maturing in the thymus to naive state in the periphery, circulating in and out of lymph nodes, activated in lymph nodes, turning into effector cells. I'll talk briefly about memory. Abul didn't talk too much about memory. Migration to the site of infection and eradication of the infection. And so it's the first step that we have to consider is you have this naive cell activated by antigen through a professional antigen presenting cell, an expanded clone, and they're in the lymph node. And some helper T cells can actually do things that are useful in the lymph node, but others are not going to be useful in the lymph node. They have to get elsewhere, and they have to get out. So what do you think of the cells that stay in the lymph node, the helper T cells, helper CD4 positive T cells, whose job it is actually to stick around in the lymph node? What do you think they're doing? So you mentioned B cells. They're helping B cells. In fact, the original definition of helper for helper T cells was that some antibody responses needed T cells to help them along. And since in the lymph node or any other secondary lymphoid organ there are follicles with B cells, some of these guys are going to stay around here and help the B cells in an antibody response. And Shiv Pillai is going to talk about how helper T cells help B cells. But if you're talking about T cells that really need to deal with infections in peripheral tissues, they have to get out. And so getting out, like everything else in trafficking of lymphocytes in immune responses, is not sort of a random process, but highly regulated. How it's regulated is by a system of expression of receptors that see a chemical that is more highly expressed in draining lymph from the lymph node rather than in the tissue within the lymph node. And that chemical is a lipid called sphingosine-1 phosphate. And sphingosine-1 phosphate is made everywhere in the body, but tissues tend to degrade it, whereas in the blood and the lymph, it's at a higher concentration. And so there's a concentration gradient from, say, this bar here would be the T cell zone of a lymph node, and the egress out into the lymph, this T cell would follow this concentration gradient, seeing where the sphingosine-1 phosphate is a higher concentration. And it does that by virtue of the fact that these lymphocytes have a receptor called sphingosine-1 phosphate receptor. And so it's this process that will bring T cells out of the lymph node. However, you can imagine if you can regulate the expression of the receptor, you can say, okay, we want a T cell to stay in the lymph node. It should have a low amount of receptors. So that is shown here on the top panel, the receptor being shown as this little green transmembrane protein here. When the receptor is expressed, there can be egress. And you might imagine you could block this with a drug, and there is actually a drug that is in clinical practice now, is approved, called fingolimod, which is approved for some cases of relapsing, remitting multiple sclerosis, that actually blocks the egress of lymphocytes from the lymph node. So just briefly, how is this receptor modulated? When a naive T cell gets into a lymph node, it has very little of the receptor present. Therefore, it won't be drawn out, and the naive T cell needs to move around in the lymph node looking for a dendritic cell that might have the antigen that it recognizes. And so the naive T cell is given a chance to find its antigen. If over time the antigen isn't found, it will be re-expressed. The sphingosine 1-phosphate receptor will be re-expressed, and the lymphocyte will get out. If the naive T cell sees the antigen and becomes activated, its receptor, the sphingosine 1-phosphate receptor, is also suppressed. It's suppressed in a complicated way by a presence of another molecule called CD69. That's a detail you don't need to know. But you want the activated T cell, the cell that's activated by antigen, also not to leave right away. Why don't you want the activated T cell? It's seen the antigen presented by the dendritic cell. Why do you want it to hang around in the lymph node? Why not just get it out of there? It's seen, say it's seen, the naive T cell's been in the lymph node for a few minutes. It sees an antigen that it recognizes. It gets signals. Why not get that lymphocyte right out? Yeah, all the things that the bull told you. You take proliferation and differentiation, that takes several days. So you don't want a only partly differentiated T cell or only a few of the T cells to get out. You want a fully clonally expanded, fully differentiated population to get out. And so again, the suppression of sphingosine 1-phosphate receptor keeps that activated T cell in long enough. So let's say that happened and that's successful, and you get this clonal expansion of CD4 positive T cells that are now differentiated. They're the ones now that have to leave the lymph node and get out into the periphery. And so that will happen. They'll get into the blood. So I don't know how many people have thought about this in this audience, but if the lymphocyte is getting out into the lymph, how does it get back into the blood? So lymph nodes basically are connected from one to another by lymphatic channels. The lymphatics drain tissues. What's the connection with the blood? So there is this general accumulation of large and large lymphatic channels into some major ones. The major one in the body is the thoracic duct, and that's where all the lymph gets dumped back into the venous blood that gets into the heart. And so this is part of lymphocyte recirculation. Naive cells go into lymph nodes. They drain from lymph. They go back into blood. They go into other lymph nodes. Naive cells that get activated turn to effector cells. They also go through the lymph, back into the blood. And now those activated effector CD4 T cells, instead of going back to other lymph nodes, will preferentially go to sites of infection where they'll do their job. Okay, so how do they actually get out of the blood vessel to a site of infection? This is similar to the general paradigm that I showed you one slide of in the innate immunity lecture, and it went by fairly quickly, where there are adhesion molecules on the endothelial cells that bind to counter receptors or adhesion molecules on the lymphocytes that allow the lymphocytes to stick. And there are chemokines made in inflammatory sites that will bind to chemokine receptors on these cells. And the combination of adhesion molecules and chemokine signaling will allow lymphocytes that are moving along in the blood vessel to stick, roll along the endothelium, stop, and then crawl through. So it's just like neutrophils and monocytes that I described in innate immunity. It's just here the chemokines are a different set of chemokines. There are specific chemokine receptors on lymphocytes that will be attracted by certain chemokines, and they're different than the ones that attract neutrophils and monocytes. It turns out that, and let me see whether I've got the right slide. I do, that blocking some of those mechanisms of moving effector lymphocytes out of the blood into the tissue is a therapeutic strategy, and one that is actually used somewhat successfully. Again, for multiple sclerosis, a drug called Tesabre, which is basically an antibody against the ligand for VCAM1, that is the integrins that adhesion molecules on the lymphocyte that bind. It blocks this interaction, and it stops T cells from actually getting through blood vessels in the brain into the CNS in patients with multiple sclerosis, and CD4 positive T cells are part of the process in that disease. Okay, so, yeah. So the question is, what causes the migration of the lymphocytes through, it could be the vessels in the brain, but it could be any tissue where you have T cells going out. And let's go back to infection, let's say to a site of infection. What causes that is the innate response locally has upregulated the expression of the chemokines and adhesion molecules, so it makes this endothelium sticky for those T cells. This will happen at any inflammatory site. It's not specific for antigen at this stage. So the antigen has nothing to do with, as far as most data shows, with the ability of a T cell to get out at this site. It has to do with these general non-antigen specific processes, that is adhesion molecules and chemokines. Okay? All right, so then that allows the movement of the lymphocyte out into the tissue. And now there's another step where the T cell has to be activated. It gets out of it was originally activated in the lymph node as a naive T cell by antigen. It's become an effector cell, has gotten in the blood. It's found an inflammatory site where the right signals are for it to get out of the blood into the tissue. But it's still not going to do any good until it sees antigen again, the exact same antigen presented now by perhaps different types of antigen-presenting cells in the tissue, where it becomes reactivated to perform the functions that are useful, that is killing the bacteria that's in the phagocyte, as an example, or expressing cytokines that will recruit more inflammatory cells. And so in the tissue, the recognition of antigen is a necessary step, and it's got to remember, it's the same peptide MHC that the effector T cell may see in the tissue as it saw way back in the lymph node. It's just in the lymph node, the antigen-presenting cell needed for the naive T cell activation is what type of cell? Dendritic cell. Now that you are an effector cell, you can be activated by all sorts of different types of antigen-presenting cells, and important ones are macrophages. If it's a CD4 positive T cell, it has to be an antigen-presenting cell that has class 2 MHC on it, and in inflamed tissue, a predominant one would be a macrophage, an activated macrophage that will express class 2 MHC. So it could be dendritic cells out in the peripheral tissue, because there are dendritic cells out there, but it can also be macrophages. And this requirement for co-stimulation that Abul talked about is so critical for naive T cell activation is not so big a deal once you're an effector cell. It's less of a hurdle. The antigen recognition is probably sufficient for some degree of activation. There may be some other co-stimulatory molecules involved at this stage that modulate or enhance, but they're not critical. Okay, so this is now an overview in this slide of sort of a generic CD4 T cell mediated response. What does the T cell do once it's out in the tissue? And the best defined way helper T cells work in classic cell-mediated immunity is illustrated here, but it's not the only way, but I'm going to put this one forward as the first example. And then when I talk about subsets, I'll refine the picture. And so you got the effector T cell out here. Here it is. It will see antigen presented by, in this case, a macrophage that has engulfed some, say, microbe, a bacteria, has processed it, taken a bacterial protein, put a peptide on a class 2 MHC molecule, and presented it now to this helper T cell, this effector CD4 positive T cell. That will allow a bidirectional communication and activation of the helper T cell and the macrophage. And when I mean bidirectional, let me explain what I mean. Antigen recognition by this T cell will cause activating signals in the T cell. So the T cell now will start expressing molecules that will work back on the macrophage. So molecules that an activated T cell can express, and in particular CD4 helper T cells, are cytokines. In this case, we're now talking about different cytokines than we talked about in innate immunity. They will also express surface molecules that are bound in the membrane but interact directly with surface molecules on the activated macrophage. And the most important one to think about here is something called CD40 ligand, which binds to CD40 on the macrophage. This pair of molecules is prominent in the adaptive immune system. It's the same pair of molecules that are the way helper T cells talk to B cells that Shiv Pillai will tell you about in the afternoon. In this case, the ligand binding to the receptor CD40 induces signals in this cell, together with cytokines that activate this cell even further than it was activated by the innate immune system here, just with the bugs. And so if you can imagine that over eons, bacteria have learned strategies to not get killed by the phagosome in a macrophage, that then a secondary event in evolution of our immune systems was, okay, that's not enough to eradicate these bugs. Now we have adaptive immunity. The T cell is now throwing these other signals to the macrophage, including cytokines and CD40 signaling, that will enhance the killing mechanisms, and the bug gets killed in a T-dependent way. In addition, these cytokines may promote further activation of other leukocytes, including further recruitment of leukocytes, because these cytokines can be inflammatory. Ultimately, the bug is killed, and over time, these effector T cells that have done their job will die by a programmed cell death or apoptosis. This part of the process is very important because we send armies of these cells in to deal with infections, but these are dangerous cells, these activated helper T cells, and we want to get rid of them once the antigen stimulus is gone. And so there's a tremendous decrease in the amount of effector cells in any immune response. This is the downregulation phase of an immune response. Of course, at the end of the day, you'll also get memory T cells that I'll talk about now. So that general scheme clear. There's this two-step antigen recognition in cell-mediated immunity, first in the lymph node, then as the effector cells in the tissue. Helper T cells, by definition, work by helping other cells, and a classic way this can be done is by helping macrophages kill ingested bugs. We'll talk about other ways in a minute when we talk about subsets. Any questions? Okay. So at the end of an immune response like I just described, there is, and that's one of the remarkable things about adaptive immunity, there is memory. And so you have a process whereby, in addition to generating a lot of effector T cells, you get long-lived cells that are not actively secreting cytokines, not actively expressing CD40 ligand, not doing all those things that the active helper T cell is doing, but they can be induced to do that much more quickly and efficiently on the second exposure or the subsequent exposure to the microbe than can a person starting from scratch with only naive T cells. One reason memory T cell responses are faster, and this is also similar for antibody responses, is there are more memory cells specific for that microbe at the end of the day than there were initially naive T cells, so that we can, and I'll go back to this slide earlier, sorry for this, that at the end of the day, the number of cells that you have specific for an infection here, microspecific T cells, is higher than what you started at the beginning. So where you might have, a few hundred cells specific for one peptide of some virus before an infection, you may have many, many thousands of them circulating around or present in your lymphoid tissues. Furthermore, the memory cell has already gone through a lot of the differentiation steps, and it's not like starting with a naive T cell. The signals needed are less and the responses are quicker. So we identify memory cells by surface markers imperfectly. We don't do a great job of that. And in humans, we use a molecule called CD45 that is a big membrane protein on T cells that it has a phosphatase activity. It's involved in T cell activation. But there are different isoforms of it. And so the RO isoform is typical of a recently activated T cell, but also it stays RO as a memory T cell, whereas RA is on naive T cells. The IL-2 receptor, which is CD25, at least one chain of it, is something that goes up quite high in effector cells, but it drops down low. But the IL-7 receptor, CD127, which is high on naive T cells, is also high on memory cells. And then there's another marker, CD44. It's actually a molecule involved in migration, but it also is a good marker for memory if it's high. So you use several different markers to define memory populations in humans, and you can largely do this out of the peripheral blood. And if you look at a newborn baby and take the blood out of an umbilical cord, it's virtually all CD45 RA positive. It looks like this. They're all naive cells. As we become more and more experienced over time and have more and more infections, more of our T cells that are in our lymph nodes and circulating in our blood become memory, and somebody as old as me has more memory cells than naive T cells in the blood. The memory can last a lifetime. It's remarkable how long these live, and there probably is a slow replacement of these cells driven by IL-7 that binds to this molecule. And so it's not like I have the same memory cells that I did 25 or 30 years ago, but it's the same clone of cells that is slowly being replenished as cells die off. And you can have memory, as you know, and some of the best examples are B-cell memory, but you can have T-cell memory that literally lasts for decades and decades. And that is something that, although there are controversial reports in the innate immune system about memory, there's virtually no effect of memory in the innate immune system. Your response to a subsequent infection, your innate response is exactly the same as the first time. That's not true for T-cell responses or antibody responses. Okay. Any questions on memory? This is all we're going to do for it. It's a huge and important topic. Of course, memory is what you're trying to get for vaccinations, and inducing good memory responses is the golden standard of vaccines. And it's also the toughest thing to do for T cells, because we're not good at getting good T cell memory by vaccines. Okay, so let's move to now a little more specifics about the CD4 positive T cells and focus our conversation on subsets that produce different cytokines. And so this idea came out as a possibility that not all CD4 helper T cells were the same, when it was already known that helper T cells helped a lot of different cells do different things. Originally defined as helping B cells make good antibody responses. Then it was understood that helper T cells were important for good macrophage activation and killing of microbes that get inside the phagocytomes of microbes. There is at least a subset of cytotoxic T lymphocyte CD8 responses that seem to require CD4 cells, so that these guys may help CTLs. And it's been known more recently than in the past that helper T cells can actually promote a lot of acute inflammation to certain types of infections. And so how can one helper T cell do all those different things? So it was proposed that maybe there are different helper T cells, and the hypothesis originally put forward by two guys named Mossman and Kaufman working at a company in California said that maybe there are differences, the cytokines they make, and they looked for the possibility that you could have clones of T cells making one set of cytokines versus others making other cytokines. And in fact, this is what they found. And they first found this way back in the 1980s in mouse T cells, but then the information was progressed to looking at human T cells, and the same ideas were found. And they were basically called Th1 and Th2 cells because they were the first and second ones of two they found. So then 20 or more years went by before anybody considered that maybe there's more than just these two types. And what was discovered, maybe, I don't know, 6 or 7 years ago, maybe it's a little longer, was that there are subsets of T cells that are not typical of these Th1 and Th2s that make another set of cytokines. And one of those cytokines was called IL-17. So illogically, they named it Th17. And it's illogical because this is not named because it makes IL-1. It's named because it was the first, and this the second. But unfortunately, this one now is named after a cytokine they make. So that's part of our inconsistency in naming things. All right, so let me give you right up front the characteristics of these three subsets, and then describe how you get them from naive T cells and some of the interesting disease-related aspects of these. So the original Th1 and Th2s, which still are Th1 and Th2s to this day, are defined by these cytokines listed here. Interferon gamma is the signature cytokine that defines Th1 cells. Th1 cells can also make some TNF, that pro-inflammatory cytokine, but it's the gamma interferon that is the classic one that defines this. Because interferon gamma, one of its great properties is the ability to activate macrophages, you might see that that's the importance of it in immune responses, macrophage activation. Interferon gamma also can provide help to B cells in making certain antibody isotypes, but I won't emphasize that now because we have new knowledge of a type of cell called a follicular helper T cell that's probably the most important for this. So macrophage activation is what I told you about in that sort of generic response, CD4 T cell response a few minutes ago. And so what are the types of microbes that this is a good defense for? It's the ones that get inside cells and stay in cells. Certain bacteria can survive in phagocytomes and fend off what the innate immune system throws at them, and this is a particularly good way of getting rid of them. Does anybody have a good guess or know what a particular type of bacterium, for instance, that might be? One where survival in phagocytomes seems to be something they've developed where we need CD4 help to get rid of them. So I think I heard TB. So Mycobacterium and Mycobacterium tuberculosis in particular is an example of one of these. There are other intracellular bacteria. There are fungi that can get into phagocytomes as well. Okay, the problem happens when Th1 cells are directed against self-tissue. They can cause all the, they can throw at the self-tissue all the things they throw at the organism and can be part of the process of autoimmune diseases with tissue damage. But also, if the microbe is clever enough even to resist the helper T cell activated macrophage, then you have persistent activation of the T cell because the antigen never goes away, and persistent activation of the macrophage. And although the bug isn't getting killed, all these inflammatory cytokines and reactive oxygen species are generated, and it ends up with chronic damage to tissues. So chronic infection leading to chronic tissue damage is a problem with Th1-mediated responses. Okay, the second one, Th2s, the classic cytokines or the signature cytokines of them are IL-4, IL-5, and IL-13. These are involved largely for a protective mechanism for dealing with helminths or worms, helminthic parasites. And they do this because they can, IL-4 can produce or activate B cells to make IgE antibody. IgE binds to mast cells and also to eosinophils. And IL-5 is a potent eosinophil developmental cytokine as well as activator. Mast cells and eosinophils granules contain things that are really toxic for bugs. These cytokines and IL-13 are involved also in the expulsion of microbes from the gut, worms that are stuck in the gut wall. It will cause peristalsis and a lot of mucus secretion. And so it's probably helminthic parasites that are this evolutionary driving force for developing a branch of our CD4 T cell immunity that makes these cytokines. The bad side of this is that IgE induced by IL-4 and eosinophils by IL-5 are potent components of allergic disease, are part of this whole range, the most common form of immunologic disease. And in societies where there are not a lot of worms, most Th2s, all they do is cause problems rather than help us, probably. Okay, and Th17 cells make IL-17. There are many flavors of IL-17. The two that are made by Th17 cells are A and F, and the cytokine IL-22. They make some other cytokines, but these are the defining ones. And the immune response that they drive is strikingly neutrophilic and monocytic. It looks like the acute inflammatory responses that I described in innate immunity. And this is a bit of a revelation when we understood this several years ago, that you can have a T cell response that's not just a bunch of macrophages and other T cells. You can have T cell responses which, when you look under the microscope or you fax, flow cytometry, look at the infiltrates, they look like innate responses filled with neutrophils, but they're driven by T cells. And you might imagine that those phagocytes are really most important for getting rid of bugs that are outside the cells and kill them. And it turns out that extracellular bacteria and fungi are the important targets for Th17-mediated immunity, and I'll show some evidence for that in a few minutes. Once again, and don't ignore the organ here, organ-specific. There's meant to be no distinction between these two. Th17 cells have been shown to be very much involved in autoimmune diseases, and in fact, in any disease that we originally thought was Th1-mediated autoimmunity, it turns out Th17 cells are there as well. And whether there's a sequential change from Th17 to Th1 over time is not clear, but that may happen. But this idea that you have solely Th1-mediated versus Th17 diseases is not true. And if you look in humans and not mice, you will actually see a lot of T cells that make both gamma interferon and IL-17. So they are neither Th1 or Th17, but some of the lessons we learned from the very pure subsets apply to these cells as well. And so the reality is that life isn't simple, and immunology isn't a dichotomy or a trichotomy, and we have variations of these. The take-home point, though, for these subsets is with chronic repetitive restimulation, such as in chronic infections or an autoimmunity where the antigen never goes away, you will drive polarization down these subsets. Or in allergies where you're constantly exposed to environmental antigens you can't get rid of, like the cat that you don't want to remove from the house because you love the cat. You will drive towards these polarized subsets. In most day-to-day immune responses in humans, CD4 T cells have a mix of cytokines, not just these classic polarized subsets. Okay, so the general principle of the CD4 subsets is they make some overlapping sets of cytokines. Early after activation, when you start with a naive T cell driving it to clonal proliferation and differentiation, there'll be many cytokines that they can produce. But with progressive activation, you'll get these polarized subsets. They not only have distinct functions that I described briefly in that table in the previous slide, but they also can migrate differently, and they have different roles in disease. So let's ask the question, how do you get one or another subset? And so the relevance of this might be if you know that allergic diseases are really underpinned by or require Th2 cells, then one way to attack treating allergic diseases, apart from the somewhat hit or miss ways we do it now, is to stop the differentiation of Th2 cells specific for an allergen. And if you know the steps in differentiation, maybe there's a way of doing that. So that's one example. Or if you know that an autoimmune disease is particularly dependent on Th17 cells, then understand the biology of differentiation. So that's sort of the driving reason for understanding these pathways. And we are starting to develop drugs like small molecule inhibitors that actually block differentiation of subsets, as well as we have biologic drugs against certain factors or cytokines that are needed for differentiation. So it turns out that the cytokines produced way back in that lymph node, when the naive T cell is seeing the antigen presented by the dendritic cell, the cytokines in that little microenvironment are the most important factors for driving pathways down, driving T cells down pathways like Th1, Th2, or Th17. And the source of these cytokines is usually the antigen-presenting cell and or cells in the environment. In the lymph node, there's not just dendritic cells around, there are stromal cells, there are various forms of macrophages. There are lots of potential sources of cytokines. And this is sort of a teleologic thing that we like to say, that the type of subset that is created at the end of the day with a particular microbe is the best one to attack or kill that type of subset. It sort of makes some theologic sense that we've evolved that a helminth would be really good at inducing Th2 responses because of what I told you about Th2s getting rid of helminths, and that mycobacterium may be really good at Th1 responses. And in some ways, if you look at this, that fits the picture. But of course, it's not always perfect. Okay, so the differentiation of these subsets from the naive T cells, you can divide into steps arbitrarily. This is a continual process in time. But you start with an induction phase. And what's key to this is that because of those signals, and I'm saying already that cytokine signals present early on that drive this, you get the expression of certain transcription factors that define the lineage, a Th1 transcription factor versus a Th2 versus a Th17. You start getting synthesis because of those transcription factors sitting on genes that then induce transcription of certain subset-specific cytokines. Then, because of repetitive stimulation and the signals generated by these cytokines, there ends up being a real commitment, and that involves not just what we call acute transcription of genes, but actual stable changes in these loci, which are done by epigenetic changes. These are not changes in gene sequence, of course, and these are, for instance, various forms of methylation of histone proteins or demethylation, changes in the epigenetics that lead to stable cytokine production. And then it turns out that these T cells themselves, as they differentiate, some of the cytokines they make work in an amplifying way. They work back on the earlier T cells in the process and reinforce or amplify a differentiation. So I'll show you this in some detail for each of these subsets, 1, 2, and 17. These, although they may look complicated, they're already simplified from what the reality is, but it gives you a general picture, and it'll be the same paradigm for each of these subsets. So you start with a naive T cell, and it sees antigen presented by a dendritic cell. If this microbe in this dendritic cell was one that had strong TLRs that bound to, strong TLR ligands that bound to TLRs, toll-like receptors in the dendritic cell, it will induce that dendritic cell to make quite a bit of IL-12, interleukin 12. It's a cytokine that is very important for Th1 differentiation. So the IL-12 will work through an IL-12 receptor and through a transcription factor that induces, called STAT4. And this cell will start making some gamma interferon. There's other sources of gamma interferon that can occur from other cells, such as natural killer cells. The combination of IL-12 and gamma interferon sets up a situation where this transcription factor called Tbet becomes dominant and stably expressed. Tbet is the master regulator or Th1-defined transcription factor for Th1 differentiation. And the gamma interferon that is made will form an amplifying loop. And you end up with Th1 cells in which interferon gamma is the main cytokine. So these are the functions I already listed to you before. So the lesson is cytokines induce transcription factors, and some of these are downstream of the cytokine receptors, and some are indirectly downstream of cytokine receptors, but they are defining the subset. And so when you look at Th1 cells, they'll all express Tbet, gamma interferon cells, and we can actually use this for interrogating infiltrates in diseased tissues. For instance, in humans, where you take the T cells out of a synovial joint and you say, what are the transcription factors expressed? Look, here are a bunch of CD4 cells that we've isolated, and they all express Tbet. That's an indicator, one indicator of Th1 differentiation. For Th2s, it's a similar type of story. In this case, IL-4 is the driving cytokine, not IL-12 or interferon gamma, that induces the differentiation of this subset to produce cytokines that include IL-4. And the defining transcription factor for this subset is GATA3. Again, GATA3 is a good marker for Th2 differentiation. They work at opposition to one another. GATA3 suppresses Tbet expression. Tbet expresses GATA3 expression. So that's part of the process of polarizing. As a cell gets committed, it makes things that will inhibit the differentiation of the other pathway. And ultimately, you get this IL-4, IL-5, IL-13 secreting T cell. Now, you might ask, where does the IL-4 come from? Since IL-4 is ultimately the product of Th2 cells, we can't depend on IL-4 being around before there's any Th2 cells in this response. And so that's been a big mystery. There are various cells that have been put forward as the source of IL-4 that's critical for Th2 differentiation, including mast cells, eosinophils. Maybe the naive T cell itself makes a little and starts the amplification process. There's various Nature papers and Nature Immunology papers and immunity papers, high-impact immunology research journals each year with a new source of IL-4 for Th2 differentiation, and then a paper that disproves it the following year. So nonetheless, we know IL-4 is critical and important for this process. And then for Th17 cells, again, are the same lessons. There are cytokines that drive differentiation, and there are transcription factors that define the differentiation. The cytokine story is a little more complicated for Th17 cells because it's really a mixture of cytokines, and actually a surprising mix. Because one cytokine that seems to be essential for this, and I think most people are happy with this both in mouse and human, is a cytokine called TGF-beta, transforming growth factor beta. And why that's surprising is, at the end of the day, Th17 cells are produced that are very inflammatory. I've told you this. They induce acute inflammation. TGF-beta has been long known as sort of an anti-inflammatory cytokine. Before a lot of the molecular basis of it was known that TGF-beta would turn down immune responses, would suppress T-cell function, and yet here that cytokine is important for generation of a very pro-inflammatory cytokine. And in fact, Bull will tell you this afternoon that TGF-beta is absolutely essential for the differentiation of regulatory T cells. And that is, again, the opposite of what a Th17 does. So how can you have this cytokine producing these inflammatory effector T cells when it has otherwise so many anti-inflammatory properties? And the answer is that it has to be in combination with these potent innate cytokines. Remember, I told you I'd return to IL-1 and IL-6 several times today. Here I am again. These are these innate response cytokines. And in fact, some people have even put TNF in this picture in some differentiation schemes. So TGF-beta plus strong innate immune response cytokines generated by some innate activation of the dendritic cell probably will drive down towards Th17s. The transcription factor that defines this is what's called ROR gamma T. That's the protein. The transcription factor is actually called, the gene is called ROR-C, so there's sometimes some confusion in the literature. But this defines the Th17 pathway. And I just want to point out that STAT3 is this transcription factor that's activated by IL-6, and as well as IL-23 that's involved in this. And I'll return to that in a minute because there's some interesting people with STAT3 mutations that I'll show you what happens with Th17 cells. So here the story is TGF-beta plus IL-1 or IL-6 leads to Th17 differentiation, ROR gamma T being the defining transcription factor. Yeah. Let's ask the question, how do you get one or another subset? And so the relevance of this might be if you know that allergic diseases are really underpinned by or require Th2 cells, then one way to attack treating allergic diseases, apart from the somewhat hit or miss ways we do it now, is to stop the differentiation of Th2 cells specific for an allergen. And if you know the steps in differentiation, maybe there's a way of doing that. So that's one example. Or if you know that an autoimmune disease is particularly dependent on Th17 cells, then understand the biology of differentiation. So that's sort of the driving reason for understanding these pathways. And we are starting to develop drugs like small molecule inhibitors that actually block differentiation of subsets, as well as we have biologic drugs against certain factors or cytokines that are needed for differentiation. So it turns out that the cytokines produced way back in that lymph node, when the naive T cell is seeing the antigen presented by the dendritic cell, the cytokines in that little microenvironment are the most important factors for driving pathways down, driving T cells down pathways like Th1, Th2, or Th17. And the source of these cytokines is usually the antigen-presenting cell and or cells in the environment. In the lymph node, there's not just dendritic cells around. There are stromal cells. There are various forms of macrophages. There are lots of potential sources of cytokines. And this is sort of a teleologic thing that we like to say, that the type of subset that is created at the end of the day with a particular microbe is the best one to attack or kill that type of subset. It sort of makes some theologic sense that we've evolved that a helminth would be really good at inducing Th2 responses because of what I told you about Th2s getting rid of helminths, and that mycobacterium may be really good at Th1 responses. And in some ways, if you look at this, that fits the picture. But of course, it's not always perfect. Okay, so the differentiation of these subsets from naive T cells, you can divide into steps arbitrarily. This is a continual process in time. But you start with an induction phase. And what's key to this is that because of those signals, and I'm saying already that cytokine signals present early on that drive this, you get the expression of certain transcription factors that define the lineage, a Th1 transcription factor versus a Th2 versus a Th17. You start getting synthesis because of those transcription factors sitting on genes that then induce transcription of certain subset-specific cytokines. Then, because of repetitive stimulation and the signals generated by these cytokines, there ends up being a real commitment. And that involves not just what we call acute transcription of genes, but actual stable changes in these loci, which are done by epigenetic changes. These are not changes in gene sequence, of course. And these are, for instance, various forms of methylation of histone proteins or demethylation, changes in the epigenetics that lead to stable cytokine production. And then it turns out that these T cells themselves, as they differentiate, some of the cytokines they make work in an amplifying way. They work back on the earlier T cells in the process and reinforce or amplify a differentiation. So I'll show you this in some detail for each of these subsets, 1, 2, and 17. These, although they may look complicated, they're already simplified from what the reality is, but it gives you a general picture. And it'll be the same paradigm for each of these subsets. So you start with a naive T cell, and it sees antigen presented by a dendritic cell. If this microbe in this dendritic cell was one that had strong TLRs that bound to, strong TLR ligands that bound to TLRs, toll-like receptors in the dendritic cell, it will induce that dendritic cell to make quite a bit of IL-12, interleukin 12. It's a cytokine that is very important for Th1 differentiation. So the IL-12 will work through an IL-12 receptor and through a transcription factor that induces called STAT4. And this cell will start making some gamma interferon. There's other sources of gamma interferon that can occur from other cells, such as natural killer cells. The combination of IL-12 and gamma interferon sets up a situation where this transcription factor called TBET becomes dominant and stably expressed. TBET is the master regulator or Th1-defined transcription factor for Th1 differentiation. And the gamma interferon that is made will form an amplifying loop. And you end up with Th1 cells in which interferon gamma is the main cytokine. So these are the functions I already listed to you before. So the lesson is cytokines induce transcription factors, and some of these are downstream of the cytokine receptors, and some are indirectly downstream of cytokine receptors, but they are defining the subset. And so when you look at Th1 cells, they'll all express TBET, gamma interferon cells. And we can actually use this for interrogating infiltrates in diseased tissues. For instance, in humans, where you take the T cells out of a synovial joint and you say, what are the transcription factors expressed? Look, here are a bunch of CD4 cells that we've isolated, and they all express TBET. That's an indicator, one indicator of Th1 differentiation. For Th2s, it's a similar type of story. In this case, IL-4 is the driving cytokine, not IL-12 or interferon gamma, that induces the differentiation of this subset to produce cytokines that include IL-4. And the defining transcription factor for this subset is GATA3. Again, GATA3 is a good marker for Th2 differentiation. They work at opposition to one another. GATA3 suppresses TBET expression. TBET expresses GATA3 expression. So that's part of the process of polarizing. As a cell gets committed, it makes things that will inhibit the differentiation of the other pathway. And ultimately, you get this IL-4, IL-5, IL-13 secreting T cell. Now, you might ask, where does the IL-4 come from? Since IL-4 is ultimately the product of Th2 cells, we can't depend on IL-4 being around before there's any Th2 cells in this response. And so that's been a big mystery. there are various cells that have been put forward as the source of IL-4 that's critical for Th2 differentiation, including mast cells, eosinophils. Maybe the naive T cell itself makes a little and starts the amplification process. There's various Nature papers and Nature Immunology papers and immunity papers, high-impact immunology research journals each year with a new source of IL-4 for Th2 differentiation, and then a paper that disproves it the following year. So nonetheless, we know IL-4 is critical and important for this process. And then for Th17 cells, again, are the same lessons. There are cytokines that drive differentiation, and there are transcription factors that define the differentiation. The cytokine story is a little more complicated for Th17 cells because it's really a mixture of cytokines, and actually a surprising mix. Because one cytokine that seems to be essential for this, and I think most people are happy with this both in mouse and human, is a cytokine called TGF-beta, transforming growth factor beta. And why that's surprising is, at the end of the day, Th17 cells are produced that are very inflammatory. I've told you this. They induce acute inflammation. TGF-beta has been long known as sort of an anti-inflammatory cytokine. Before a lot of the molecular basis of it was known that TGF-beta would turn down immune responses, would suppress T cell function. And yet here, that cytokine is important for generation of a very pro-inflammatory cytokine. And in fact, Boole will tell you this afternoon that TGF-beta is absolutely essential for the differentiation of regulatory T cells. And that is, again, the opposite of what a Th17 does. So how can you have this cytokine producing these inflammatory effector T cells when it has otherwise so many anti-inflammatory properties? And the answer is that it has to be in combination with these potent innate cytokines. Remember, I told you I'd return to IL-1 and IL-6 several times today. Here I am again. These are these innate response cytokines. And in fact, some people have even put TNF in this picture in some differentiation schemes. So TGF-beta plus strong innate immune response cytokines generated by some innate activation of the dendritic cell probably will drive down towards Th17s. The transcription factor that defines this is what's called ROR gamma T. That's the protein. The transcription factor is actually called, the gene is called ROR-C, so there's sometimes some confusion in the literature. But this defines the Th17 pathway. And I just want to point out that STAT3 is this transcription factor that's activated by IL-6, and as well as IL-23 that's involved in this. And I'll return to that in a minute because there's some interesting people with STAT3 mutations that I'll show you what happens with Th17 cells. So here the story is TGF-beta plus IL-1 or IL-6 leads to Th17 differentiation, ROR gamma T being the defining transcription factor. Yeah? Yeah, that's an excellent question with not an absolutely clear-cut answer, but there is clearly evidence that you can get some of these epigenetics changes are maintained in the memory cells, and the memory cells on response will be potent IL-17 producers or gamma interferon producers after response. So in general, people think yes, but it's still a question being pursued, and there may be some backtracking in differentiation, but they rapidly reattain that phenotype with the right stimuli. But because the epigenetic changes are there, and apparently they're maintained over time in memory, they haven't. What I don't know is whether a Th17 cell, memory cell exists 25 years later after a Th17 response. And a lot of these, again, these studies have done in mice, and it's not clear about in humans. Okay, so let's just look briefly at the functions of these cells in a little more detail. And some of this will be reviewed for Th1 and a little bit for Th2, but Th17 I really haven't talked about function much at all. And so the Th1 function, which is at this point, here's your effector T cells. Gamma interferon defines mainly what the function is. And for macrophages, it's the activation of a macrophage to get to be a better, basically, killer of microbes. It's a very pro-inflammatory type of macrophage. And the activation of a macrophage by gamma interferon, largely from Th1 cells, is referred to as classical macrophage activation. And there's a emerging, or actually it's a long-term literature, but now an emerging interest in subsets of macrophages. And there are things called M1 and M2 macrophages. And the classically activated macrophage is the typical M1 macrophage, one that is very pro-inflammatory and very much microbiocidal. It's also very good at killing tumor cells and various other functions. As I pointed out, the cytokines made by Th1 cells can make B cells switch to antibodies, but we'll relegate that to a fairly newly defined subset called T follicular helper cells. And there may be Th1-like T follicular helper cells. Shiv will make this more clear to you. They may be derived from Th1 cells, but let's not think of that as a primary function of Th1 cells now. Okay, so let's look at the back in detail at the Th1 macrophage interaction. The 40 ligand binding to the 40, which generates signals that enhance killing. The cytokines, and in this case now I fill in from that slide I showed before, the cytokine is gamma interferon, so gamma interferon receptor signaling. What they do is upregulate the expression of enzymes that I talked to you briefly about when I talked about innate immunity that generate free radicals that are very bad for bugs, also bad for our own tissues, reactive oxygen species, nitric oxide. These signals of gamma interferon and CD40 increase the secretion of inflammatory cytokines, TNF, IL-1, IL-12, chemokines. All that will enhance more inflammation, will bring more leukocytes into the site where the infection is. And they'll also upregulate co-stimulatory molecules in class 2 MHC, which will make these cells better antigen-presenting cells. I don't know what the importance of B7 is at this point. As I said, it's not really critical to activate effector T cells, but class 2 is important for reactivating more CD4 positive T cells. So what happens when this process goes on and on? So we referred to Mycobacterium tuberculosis, and I said they evade innate immune killing in bugs, but in fact some Mycobacterium tuberculosis doesn't get killed off even when CD4 T cells are thrown at it. And that's not true for most patients infected with MTB. In fact, most people do retain and clear the infection and don't get chronic tuberculosis, but a subset do, and as we know, it's a huge clinical problem in various parts of the world. And so what's happening there is a Th1 response that never gets turned off because, in fact, the bug gets sequestered even though there's reactive oxygen species and proteolytic enzymes thrown at it. It keeps on living so that the T cell and the macrophage keep talking to each other, and you get these basically these spherules of growing macrophages, not growing, but activated macrophages, some of them fusing together to form giant cells, surrounded by activated CD4 positive T cells, basically a local cytokine storm, if you will, and you end up with a lot of cytokines that are changing the tissue around it with a lot of chronic inflammation, and you get the typical granuloma. So this is granulomatous disease, and this is basically Th1 process that isn't turned off. So there are other granulomatous diseases that are probably Th1 driven for which we don't know the antigens, like sarcoidosis, which is a mystery antigen, but it's the same general process. The granuloma look a little different, but you can think of that as basically CD4 Th1 mediated process. Chronic granulomatous disease. That's an interesting story in a way. It's similar because of the lack of, and there's an innate immune defect, the lack of being able to generate the reactive oxygen species. Certain bugs aren't killed, and so the same thing is chronic stimulation, and you get CD4 T cells involved. So that's true. There's another good example. Oh, there's examples in CTL-mediated killing that are defective that maybe Shiv will talk about with perforin knockouts. And there it's not Th1s, but it's the same thing. Gamma interferon is produced chronically because you can't kill the virus, for instance. Okay, so ultimately you get a lot of tissue remodeling and damage, and so this is a terrible-looking lung here with granulomas all filled with it. These are these giant cells here, which are basically activated macrophages with fused nuclei. And in TB, you get central necrosis, and then all of a sudden you'll get acute inflammation back here because necrotic tissue induces a strong innate response. Okay, the functions of Th2 cells we talked about. Again, IL-4 is an IgE switch factor. Again, it may be a T follicular helper cell version of Th2 cells that does this, that Shiv will talk about. But the IL-4 and IL-13 are important for expulsion of intestinal parasites. But an interesting other component is that these cytokines actually work on macrophages to activate them in a different way than gamma interferon does. And it's something that we refer to as alternative macrophage activation, where those macrophages aren't terrific at killing microbes, but they are really good at inducing tissue remodeling. In particular, they make angiogenic factors and factors that activate fibroblasts to make collagen. And so alternatively activated macrophages by chronic Th2 responses are considered part of the pathology of certain fibrosing diseases, in particular in lung. And so we have this dichotomy of macrophage activation, which simplistically, I'll look through the lens of a helper T cell biologist, can be a Th1-driven M1 macrophage. These are the things I told you that Th1-driven macrophages do. Inflammation, killing of bugs. Whereas Th2-driven macrophages, alternatively activated, sometimes called M2, will induce growth factors that cause tissue repair, but also fibrosis. They also make cytokines like IL-10, and remember TGF-beta now here I'm referring to as an anti-inflammatory cytokine, that will actually suppress inflammation. So this may be a method of sort of turning down inflammation during the resolution phase of some Th1 response, but it's not clear that Th1 moves to Th2 in any reliable way in responses. So this sort of the pathologic view of this is one as getting the most interest. And largely in lung, I think, but there may be other places where Th2s drive inflammation, tissue repair and fibrosis. So there are common misconceptions about Th1, Th2 subsets, and maybe now Th17 subsets. One is that Th1 cells are cell-mediated immunity and Th2 are humoral immunity. Really, the modern way of expressing why this is a misconception is probably neither of those are the relevant things to think about for humoral immunity. The point is, though, that Th2 cells have no particular special place in antibody-mediated help. They make IL-4 that's good for IgE switching, but gamma interferon made by Th1 cells is a perfectly good antibody switch factor that induces certain types of antibodies. So you'll read in the literature, Th1 cell-mediated immunity, Th2 antibody-mediated immunity. It's just wrong. Now that we know about T follicular helper cells, which again you'll hear about this afternoon, you shouldn't even think of either of these as fundamentally important in humoral immunity. You have to think of follicular helper cells. Another misconception was that these subsets don't exist in humans, and they really do, and you just have to look in the right way. You have to look at places with chronic immune stimulation. That being said, it is true that when you take T cells out of a lot of diseased tissues and you ask what cytokines they make, there may be a good Th1 subset, there may be a good Th17 subset, and there may be some that make both gamma and interferon at the same time. Rarely will you see a lot of Th2 cells present at a site where there's also Th1 or Th17. Okay, so now let's revisit the function of Th17 cells in a little more detail. These are the newer subset, relatively more recently defined and discovered. And so where the curiosity about the possibility of another subset came out of was the fact that over 20 years of doing Th1, Th2 type research or viewing the immunologic landscape just by these two T subsets, it became clear that there were many inflammatory diseases that were attributed to the gamma interferon producing Th1 cells that were actually worsened, at least in animal models, when you eliminated gamma interferon. And that didn't make any sense. And then it was shown that those diseases actually seemed to be dependent on a cytokine that was called IL-23. And it's related to IL-12. IL-12, remember, drives Th1 differentiation, but IL-23 does not. And it then was learned with a lot of work, summarized in one sentence, that IL-23 actually is important for the development of this subset that produces IL-17. And so what does IL-17 actually do? What does IL-22 do? IL-17 binds to IL-17 receptors that are ubiquitously expressed on hematopoietically derived cells like leukocytes, but also on tissue cells. And in both cases, it provides a very potent pro-inflammatory stimulus. It induces the expression of chemokines, TNF, IL-1, IL-6, factors called colony-stimulating factors, or CSFs, that drive more bone marrow production of neutrophils and monocytes. And so all of this is very pro-inflammatory, and it's why you end up getting a lot of neutrophils in places where IL-17 T cells are present, or Th17 cells. IL-17 also induces in epithelial cells, such as gut cells, gut epithelial cells, or bronchial epithelial cells, or even skin, the production of those antimicrobial peptides I mentioned to you in the very first lecture, innate lecture. And so it's important for what we call epithelial barrier immunity. It makes those barriers, like the skin and the gut epithelial cells, better at resisting penetration by commensal organisms. So it turns out that IL-22's main function is not inflammatory, but also to enhance this integrity of the epithelial barriers. And so IL-22 is a strong inducer of those antimicrobial peptides. Remember, I called them defensins. It's a potent inducer of them in gut epithelial cells. Also, IL-2 will drive the proliferation of epithelial cells in these barriers to replicate and therefore repair defects. So the actual physical integrity, the tight junctions between the cells here, the replacement of dead cells, seems to be mediated by IL-22. So you can imagine if you have an infection and you have damage, that implied infection, commensals have gotten through a breach in the barrier, or a pathogen has, you have a Th17 response. You have to repair the wall. You have to repair the damaged wall, and IL-22 is important for that. Okay, so here's a summary of the whole process: Th1, Th2, Th17 differentiation. These are the cytokines that drive the differentiation down these pathways. These are the master regulators, T-bet, gamma-3, or gamma-3, and they're the ultimate cytokines. So you can look at the slides that you'll have available as a quick summary. Yeah?
Question. Is it known that invariant NKT cells they produce IL-17?
So
Any of the invariant NKT cells?
yes. So the question was invariant or iNKT cells, and I have avoided them. I think Shiv is going to talk about them a little bit. But yes, there are definitely, so that's a very important point. I'm glad you asked it. IL-17 clearly can be made by other cells besides Th17 cells. iNKT cells are one of them. Another that turns out to be perhaps remarkably important in skin and in human skin, and maybe related to psoriasis, is something called gamma delta cells, which are almost like invariant NKT cells, meaning they have very limited antigen receptor diversity. They're almost like an innate cell. And they make IL-17, and there are a lot of gamma delta T cells in the skins of psoriatic patients. There's this other group of cells that we refer to briefly called innate lymphoid cells. They're not T cells. They don't have T cell receptors on them, but they look like either Th1 or Th17 or even Th2 cells. In other words, there's subsets of them. And so there's an IL-17 subset of innate lymphoid cells. The problem we have with these innate lymphoid cells is we have no idea how much of the IL-17 you may get in some responses due to them versus the Th17 cells. And it's very hard to do this for reasons that it's hard to explain now, but experimentally you can't get rid of them easily. There's no way of selectively depleting them. Okay, so Abul is going to spend a lot of time in regular T cells. I just want you to remember that that's another subset of CD4 positive T cells, and some of them actually are derived from naive T cells by a different differentiation pathway. And I referred to the fact that TGF-beta is involved in both the Th17 differentiation and the regulatory T cell differentiation. And that is a little scary because if you're trying to intentionally get a lot of these guys, it doesn't take much to get the right factors that will end up with a lot of these guys. So if you're trying to, in some therapeutic way, enhance regulatory T cells, you could see that if you're not knowing what you're doing, like we really don't yet, you may induce Th17 cells. Then there's another question that comes up, which is, can these guys switch from one to another phenotype? And I'll say, I actually don't know if the slide's in here, because this is a slightly older set than I meant to have here. But there is clear evidence in vitro that you can go from regulatory T cells to Th17 cells. And so that is a dangerous thing to think about, because you go from something that's suppressing immune responses to something that is a very pro-inflammatory immune response. And so there is some plasticity in these subsets. And if you look at the molecular genetic basis of this, you can see that the epigenetic changes can permit some switching with the right cytokine signals. So the role of these subsets in diseases, I mean, I think everybody's convinced that these have some important role as these chronic diseases occur. In autoimmunity, Th1 cells, gamma interferon Th1 cells have been shown certainly in mouse models and human tissues as well. The role of Th2 cells in allergy is quite well established. Th17 cells have been now filling in the gaps for what we don't quite understand about a lot of these autoimmune diseases, and Th17 cells have been implied to be present in MS, inflammatory bowel disease, rheumatoid arthritis. And so they seem to be, these guys seem to be somewhat ubiquitous in these chronic inflammatory diseases. Now, what is the actual proof for in mice? You can say, okay, let's knock out the IL-17 gene, and the mice more susceptible to some sort of infection, or knock out gamma interferon or IL-12 needed to make gamma interferon producing cells. And you can do these studies, and they all have shown us the importance of these different roles of these subsets that I've showed you in the summary slides. In humans, there are examples, rare examples. There are mutations affecting IL-12 and interferon gamma receptors or signaling, and there are defective Th1 responses in these patients, and they do get infections, for instance, with atypical mycobacterial species, which normally we can handle quite well, and these patients can't. So that's sort of a living proof of the importance of this pathway for defense against these intracellular microbes. Turns out that mutations affecting Th17 development, or IL-17, lead to a fascinating disease called mucocutaneous candidiasis and bacterial abscesses. It's really a disease that's given the name Job syndrome. It has a confusing name, sometimes called hyper-IgE syndrome, and the high IgE I'm going to ignore right now because that's not part of the immunologic deficiency here. And so one of the defects, remember I mentioned STAT3, which is a cytokine that is involved in Th17 differentiation. So many of the Job's patients who, have a mutation, one of many mutations in STAT3, and they can't make Th17 cells. And so the types of infections they get susceptible to are staph abscesses and candida in their mucosal membranes, and it shows, again, living proof that you need Th17 cells for defense against these extracellular pathogens. So now you can think about all the cytokines and the T cells that make them, and you can see that there's lots of potential strategies to block the differentiation of these effector subsets or actually block the cytokines they make. You know, TNF antagonists, the TNF is made by all sorts of innate cells, but it's also made by Th1 cells and Th17 cells. When you block TNF or IL-6 or IL-1, you actually may even be blocking Th17 differentiation. There are antibodies against this cytokine, IL-12, that actually has a subunit that's shared by both IL-12 and IL-23. And so IL-12, I told you, was needed for Th1 differentiation. IL-23, I told you, was needed for Th17 differentiation. So an antibody against this subunit of IL-12 is also an anti-IL-23 antibody, and therefore it's a potential use for any disease where Th1 and Th17 cells are prominent. As I told you, a lot of autoimmune diseases in humans seem to have both types of cells present. We know from advanced clinical trials that IL-17 targeting is very effective in reducing the severity of psoriasis. And again, it was by a good question from some of the audience, is the IL-17 coming from Th17 cells? Well, maybe, but it also could be coming from gamma delta T cells. Nonetheless, that's an important target. Treating asthma by blocking the cytokines that drive IgE production by B cells, that is the Th2 cytokines, is something that is a therapeutic strategy. So if we look down at these cytokines, a lot of these have T cell sources, and a lot of the drugs are already out there that are being used, and they may have effects that are not just exclusively from the Th subsets, but the Th subsets are on the list of their targets. So I'm going to stop there, and I have no idea how badly I went into your lunchtime. A little bit. I added a few more minutes to the delay that Abul gave me. So suggest if there are questions, come up afterwards so that... That was 4 minutes late.